Welcome to your video on section 4.9 on cell division. Before we get into cell division, I forgot to play a video for you on translation from our previous section. So we're going to do that first.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA, carrying its amino acid, binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
So now that we got a good review of translation, let's move into cell division. And here are your learning objectives. Let's talk about mitosis first. Mitosis is cell division that is occurring within our somatic cells. These are basically all other types of cells within our body except for the sex cells. The sex cells will be important in cell division called meiosis, in which it is either our oocyte, the egg, or the sperm cell. And when we talk about our somatic cell division, that means one of our cells is going to divide to produce two daughter cells. We need this for our body's development, tissue growth, and replacement of old cells and any type of tissue repair that we might need due to injury. So now we're going to revisit those centrosomes that we talked about in our previous sections. These are a pair of cylindrical centrioles. Remember, by itself it's called a centriole, but together they are called a centrosome. And what they do is they go to the polar ends of a cell and they help to organize the microtubules that facilitate these chromosome movements. Let's talk about our forms of DNA within a cell as well. So our nucleus, we said, has 46 DNA molecules. They are loosely coiled as chromatin, or they can be tightly coiled as in chromosomes, that X that we are used to seeing. And now let's move on to our cell cycle. Our cell cycle shows us our steps for replication of somatic cells, and we divide it into two identical daughter cells when we go through this process. We've got two major phases within our cell cycle. We have the interphase, which is represented by this large blue arrow, and then we have the meiotic or M phase. Our interphase is going to last approximately 23 hours, and the mitotic phase is going to last about an hour. Now let's go through each of these phases. Our interphase gives our cell time to prepare for our cell division. Our DNA is now in the loosely coiled chromatin, and we can subdivide interphase into 3 separate phases: G1 phase, S phase, and G2. In our G1 phase, we're going to see growth and production of new organelles, structures needed for DNA replication will be formed, and replication of the centrioles to produce 2 pairs. In our S phase, our DNA is going to be replicated so that we have 46 double helix strands. In order to do this, we need an enzyme, and that is going to be our DNA polymerase. All of this is going to take place in our nucleoplasm of our nucleus. So within our S phase, let's talk about the steps of DNA replication. First, we need to unwind our DNA molecule. We're going to break apart these parent strands, and we get an assembly of new DNA strands. And by creating these new DNA strands, we get restoration of the DNA double helix. This process continues until both of the strands are replicated, and these new replicated strands are called sister chromatids, and they will be attached in an area known as the centromere. So let me go back to show you. Here's our chromosome. At the center, we see the centromere. After that, we're going to enter the G phase. This is a very brief phase, and our centriole replication will be completed, and our enzymes for cell division will be synthesized and ready to go. So here is interphase. We see our synthesis of our cellular components are taking place, and we've got synthesis of DNA that'll occur in the S phase, and our duplication of those centrioles. Now let's move into our M phase, or mitotic phase. This is going to follow interphase. We've got mitosis and metaphase in order to produce 2 new cells. And the processes that will take place is mitosis, where we have division of the nucleus, and the other process is cytokinesis, where we have division of the cytoplasm. In our phases of mitosis, we will have these 4 consecutive phases take place: prophase, metaphase, anaphase, and telophase. So let's take a look at prophase first. We see that our DNA has been supercoiled into chromosomes with their sister chromatids, and our nucleolus is going to be broken down. Our microtubules, or the spindle fibers, are going to grow from these centrioles, and our centriole pairs are going to be pushed apart to the opposite poles. Dissolution of our nuclear envelope is going to take place as well. Now for our metaphase. This is our second stage of mitosis, and we can see that our chromosomes are aligned into what's known as the equatorial plate of our cell, so right down the center. And our spindle fibers are going to extend from the centrioles and attach to the centromere of each of these chromosomes. Now for the third stage, anaphase, we're going to start the spindle fibers by pulling these sister chromatids apart toward the poles. Each of the chromatids here are now a chromosome of 1 DNA double helix with its own centromere. And you could see this taking place down here as well. These are representative of the chromosomes. Here are the spindle fibers attaching and being pulled toward the opposite poles. And now for telophase. We have the arrival of the chromosomes at each pole, and they begin to uncoil and return back to the chromatin. We get a new nucleolus that is formed in each cell, and our mitotic spindles are broken up. You'll also see a new nuclear envelope is forming in each of these cells around the chromosomes, and this is the end of our nuclear division. Now we have to go through cytokinesis, which is the division of the cytoplasm between our two new cells. So I like this illustration because it shows the difference between our telophase, whereas in cytokinesis we get an actual splitting of the cytoplasm. We will see that the cytokinesis may overlap with anaphase and telophase, and we'll get a ring of a microfilament proteins at the cell's periphery. We pinch the mother cell into two separate cells, and as a result, we'll see that cleavage furrow